Welcome back to the deep dive. Today, we are taking the ultimate shortcut, right inside the cell. For decades, researchers have been, masters at mapping the vast cellular landscape of a tissue identifying B cells, T cells, tumor clusters.
The big picture stuff.
Exactly. But when you want to look at the subcellular level, what's happening inside a single mitochondrion, or how DNA repair proteins are interacting with the nucleus, that's where the technology has, well, it's hit a wall.
Yeah, it's the ultimate resolution puzzle. Our source material today details a really foundational breakthrough, not just in biology, but in computational imaging.
And it's all centered on a tool called imaging mass cytometry.
Or IMC, right. And what makes IMC such a game changer is its ability to track dozens of proteins at once, a really high level of multiplexing. It does this using antibodies tagged with heavy metal isotopes, which are then detected by mass spectrometry.
And the key advantage there is what IMC doesn't use.
Light. Exactly. Because it sidesteps traditional light microscopy, IMC just eliminates the two massive technical headaches that plague methods like immunofluorescence, or IF.
Okay, what are those?
First is signal interference from autofluorescence. That's the glow that a lot of biological tissues just naturally emit. And second, cycling artifacts, which happen when you have to repeatedly bleach or remove antibody layers to see new things. IMC is a clean, one-shot staining workflow. You do it once.
So we get clean data, high multiplexity, reliable tracking of dozens of targets at once. But there was this critical, unavoidable ceiling to classic IMC that kept us out of that nanometer world. What was that architectural limitation?
It was what we can call the spatial resolution cliff.
The resolution cliff.
Classic IMC relies on a laser that ablates or vaporizes a specific spot on the tissue, and that spot, your pixel size, is fixed at 1 micrometer, 1 ermine.
Okay, and for perspective, a human hair is about 100 micrometers wide. A cell nucleus might be 5 or 10.
Right, and if you were trying to capture detail, that size matters.
It fundamentally does.
And that's because of the Nyquist limit. Think of it as a rule of thumb for digital imaging.
Okay.
To accurately capture any structure, your resolution has to be at least half the size of the smallest feature you want to see.
So if I want to image something that's, say, 2 micrometers across, my resolution needs to be 1 micrometer, which for the nucleus works fine.
Right. Standard IMC at 1 micrometer can resolve the whole cell and its nucleus, no problem.
No.
But so many of the most fascinating structures, the engine room of the cell, are submicrometer.
We're talking about things like individual mitochondria.
Mitochondria, which are often smaller than 1 micrometer, and the crucial dense components inside the nucleus, the nucleoli.
So with that 1 micrometer resolution,
They don't appear as distinct structures. They're just pixelated, aggregated smudges, or even worse, they're completely invisible.
So the goal is set. Keep the amazing multiplexing of IMC, but somehow push that 1 micrometer barrier down to the nanometer scale. And do it without asking every lab to buy a brand new $1 million piece of hardware. And this is where our focus enters. High-Resolution IMC, or HR-IMC. They cracked this using pure computational smarts, achieving resolution below 350 nanometers.
It's incredible.
Okay, let's unpack the mechanism. This truly feels like computational magic.
It really is a phenomenal example of engineering around a physical limitation. I mean, physically shrinking the laser spot to submicrometer sizes is really, really difficult.
Oh, why is that?
You lose laser stability, you lose tissue penetration depth, and you start to introduce thermal degradation. You're basically cooking the sample.
Not ideal.
Not ideal. So HR-IMC it sidesteps this entire problem. It uses the standard laser, but it changes the process in two key steps.
Okay, 2 steps.
First, how the data is acquired. That's the oversampling. And second, how it's all mathematically corrected. That's the custom deconvolution.
Okay, let's start with that acquisition trick. Oversampling. It sounds almost counterintuitive, like they are purposefully blurring the image to start with.
They are, but it's a very purposeful blur. Instead of moving 1 micrometer after every laser shot, which gives you these nice, neat, non-overlapping pixels,
Right.
HR-IMC uses that standard 1 micrometer spot, but samples the tissue at a submicrometer step size. They tried it with 500 millimeter or 333 millimeter steps.
So if the laser spot is 1,000 nanometers wide, but they only step forward 333 nanometers, the laser shots are heavily overlapping each other.
Precisely. You get these partially redundant overlapping ablation areas. A good analogy is like taking a shovel that's 1 meter wide. But instead of taking a full step before your next scoop, you only shuffle forward about 30 centimeters.
You end up hitting the same ground multiple times.
Exactly.
Yeah.
And that overlap is the key.
Yeah.
By using a 333 millimeter step size, that 1 micrometer pixel area is effectively carved up into multiple distinct analysis points or subpixels, in this case, 9 of them.
I can see the geometry, but you mentioned a major challenge earlier. IMC is a destructive technique, so if the laser hits the same tissue repeatedly because of that overlap, doesn't the signal just vanish instantly?
That is the unique constraint IMC has that light microscopy doesn't. And it required some really sophisticated modeling to get around.
So what did they do?
To counteract that instant destruction, they had to drastically reduce the laser energy per pass. They found that a -10 dB laser energy setting was the sweet spot.
Okay, so a tiny amount of energy.
A tiny amount. It allowed the underlying signal in that region to actually survive multiple overlapping passes. They're sort of chipping away at the tissue, not blasting it.
Right, but that creates a new variable, doesn't it? The tissue that was already hit by a previous shot is going to contribute less signal than fresh tissue.
Exactly. The signal decays with every single pass. So to make the math work, they had to create a predictive model for this signal loss. They described it using an inverse sigmoidal loss function.
Okay, wait, let's slow down on that term. An inverse sigmoidal function sounds incredibly dense. Can you break that down?
For sure. Just think of it like trying to remove a stain from a shirt.
Yeah.
First time you scrub it, you remove the most material, right?
Yeah, of course.
The 5th time you scrub, you're removing very little because most of it is already gone. Your effort becomes less and less effective.
Diminishing returns.
Exactly. That sigmoidal function models this nonlinear diminishing return destruction. It accounts for the fact that a subpixel hit 3 times will have X amount of signal left, while one hit only once will have Y. And that decay isn't a straight line.
So this model lets the computer know exactly how much signal to expect from any partially ablated area.
That's the one. It's a huge computational leap. They modeled the physical destruction.
Which brings us to the second part, the deconvolution, the math fix that removes that purposeful blur.
Right. The raw oversample data is inherently blurry. I mean, every 1 micrometer shot mixes the signal from its central point with signals from all those neighboring, partially ablated subpixels.
So deconvolution has to untangle that mess.
It untangles it. It mathematically extracts the signal that came only from the precise subpixel you're interested in.
The usual tool for deblurring in imaging is something called the point spread function, or PSF. It's basically the mathematical fingerprint of the blur that's introduced by the machine itself.
Correct. The researchers used PSF-based deconvolution, specifically an algorithm called Richardson-Lucy, which is really robust against the kind of noise you find in IMC data.
But they couldn't just use a standard PSF, could they?
No, because IMC is destructive and it overlaps. This meant they needed a completely custom PSF.
So they had to invent a completely new type of deblurring software just for this machine because they were intentionally destroying the sample.
Essentially, yes. This custom PSF had to incorporate those 2 unique factors we just talked about. First, the geometrical overlap. They calculated the proportional area covered by each bordering subpixel using intersecting circle geometry.
And second,
they incorporated that inverse sigmoidal model of the expected signal remaining, given how many destructive laser passes that subpixel had already seen.
that is just brilliant. It's so complex. They accounted for both the geometry of the overlap and the material destruction rate. And by mathematically crunching that custom function over and over for every overlapping shot, they transform these low-resolution measurements into crisp, high-resolution data.
That is the key takeaway from the technical section. HR-IMC uses existing IMC hardware, but it changes the acquisition setup oversampling at reduced energy, and then applies this sophisticated custom mathematical correction to turn blurry, low-res data into clear, high-res images.
Breaking that 1 micrometer barrier to get below 350 millimeter, it really democratizes high-resolution, highly multiplexed imaging.
Absolutely.
The mechanics are incredible, but the real test is whether this increased resolution actually helps us see things that matter. How did they validate this? What did they compare it to?
They went for the gold standard. Routine immunofluorescence, or IF microscopy, which is known for its high spatial resolution. They took the exact same section of tissue and imaged it first with high-resolution IF, and then with HR-IMC, measured at 333 nanometers.
And the result?
The HR-IMC images captured details just as effectively as the IF benchmark. It was proof that the computational fix genuinely delivered that sub-micrometer resolution.
So what were they suddenly able to see that was just a 1 micrometer smudge before?
They moved from seeing these, aggregated signals to seeing true architecture. For the first time using IMC, they could clearly visualize complex mitochondrial networks.
The power grids of the cell.
The power grids of the cell, exactly, stained with markers like ATP5A. These networks were visible wrapping around the nuclei and stretching out into the cytoplasm. It's the difference between seeing a blurry street lamp and seeing the intricate electrical grid that powers a whole neighborhood.
And what about inside the nucleus?
There, they could clearly delineate nucleolus-resembling structures, which are vital for ribosome production. And crucially, they saw sharp, distinct Ki-67 foci in interphase cells.
And Ki-67 is a proliferation marker, right?
It is. Before, that signal was just a uniform blob inside the nucleus. Now, they can see specific active areas of proliferation. It gives you a much richer understanding of a cell's division state. They also showed much sharper filamentous SMA fibers, part of the cytoskeleton, which allowed for better structural separation.
That's a phenomenal biological yield. You're moving from just seeing the cell to seeing its organelles, its internal signaling architecture. But the benefit goes beyond just, pretty pictures. It has a profound practical impact on analyzing crowded tissue samples.
Absolutely. And this is where HR-IMC solves a really common failure point in immunophenotyping. The source highlights the tonsil as a perfect example.
A tonsil.
It's a very, very densely packed tissue full of small interacting immune cells.
So the technical difficulty there is that when a B cell, which is CD20 positive, and a T cell, CD3 positive, are crammed right next to each other, a 1 micrometer laser shot is going to hit both of them simultaneously.
Exactly. And classic 1 micrometer IMC averages those two signals, resulting in this impossible hybrid cell artifact they call a B and T cell.
Which is just a flaw in your analysis.
It's a fundamental flaw. If your platform misidentifies 3 adjacent cells as one giant hybrid cell, your entire quantitative analysis of that immune microenvironment is worthless.
So how much did HR-IMC actually help in this real-world scenario?
Dramatically. The high-resolution capability allowed for precise segmentation of those adjacent cell membranes and nuclei. Compared to classical IMC, HR-IMC reduced the proportion of these merged B and T cells by over 3-fold.
Wow, 3-fold.
And they confirmed this accuracy against H&E staining the histological ground truth and found HR-IMC accurately separated individual cells where the classical method failed. This practical application alone is transformative for immune-oncology studies.
So to recap, HR-IMC is validated against light microscopy. It reveals these submicrometer structures, and this high resolution translates directly into accurately counting individual immune cells in dense tissue. Now let's jump to the ultimate question. What does this level of detail teach us about a complex disease, specifically about cancer therapy?
For this, they applied HR-IMC to patient-derived high-grade serous ovarian cancer, or HGSOC.
Which is an extremely aggressive tumor type.
Extremely. They grew these cells in 3D cultures to mimic the natural environment and then used a 25-plex antibody panel. And this panel wasn't just about cell type. It targeted subcellular structures and critical markers of chemotherapy response.
Like DNA repair, apoptosis.
DNA repair, apoptosis, drug transport, all of it. This wasn't just descriptive imaging. It was functional imaging.
And what did that high-resolution view allow them to do with the data?
They shifted the entire analysis paradigm from looking at whole cells to looking at individual pixels. They used a sophisticated machine learning technique called SLOANSM to cluster individual pixels based on their unique shared protein profiles. And this identified 13 distinct subcellular regions within the cancer cells.
That is so powerful. They're literally mapping the cell's internal geography. They're not just saying this cell has high ATP5A. They're saying this specific cluster of pixels representing a mitochondrial region is defined by high ATP5A and HSP10.
Precisely. And the power is in the enhanced separation. Low-resolution data just mashes all those signals together, but HR-IMC achieved superior separation of these clusters. Cluster 4, for instance, was cleanly defined as a mitochondrial region, and Cluster 8 corresponded exactly to those Ki-67 proliferation foci we discussed earlier.
That's it.
Being able to spatially define these microregions is the necessary first step to understanding the signaling that happens within them.
So when they tracked the drug response, treating the HGSOC cells with standard chemotherapy, carboplatin, and paclitaxel, what did the high-res view confirm?
It confirmed all the expected biologically consistent shifts, but with this unprecedented spatial clarity. They saw a massive upregulation of DNA damage and repair, foci like PH2AX clusters. and a significant downregulation of proliferative clusters.
So the therapy was clearly inducing DNA damage and shutting down cell division, which is what you'd hope to see.
Right.
But the real insight comes from the subtle, hidden interactions that low-resolution imaging would miss entirely. What was the aha moment here regarding the molecular colocalization?
The ability to see where the DNA damage was actually happening. They detected a significant gain in the colocalization of phosphorylated histone H2AX. That's the DNA damage marker.
Okay.
Specifically in regions that were marked by histone methylation and acetylation variants.
And those variants mark areas of open transcription.
Exactly, where the genome is active and being read. So they didn't just see DNA damage, they saw that the cell's damage response was occurring exactly where the genome was most active, which suggests these immediate localized repair kinetics. It's a fundamentally new view of cell response.
Absolutely. And they took this discovery capability out of the 3D culture and applied it to real native ovarian tumor tissue in situ.
Mapping those same subcellular features across different cell types, tumor cells, immune cells, fibroblasts, all side by side in their natural context.
And what did that organelle-level comparison tell us about the different cell types in the tumor microenvironment?
They found that tumor cells displayed a higher mitochondrial density and larger, more interconnected mitochondrial networks than the neighboring fibroblasts or immune cells.
So this confirms known metabolic differences, but now you can actually see the architecture underlying that difference.
Right. And crucially, they were able to correlate specific metabolic states with organelle content.
You mean they found the Warburg effect happening right before their eyes?
They did. They identified hypoxic tumor cells, so cells starving for oxygen, marked by CAIX expression, and those specific cells showed a corresponding reduced mitochondrial content and a simultaneous increased expression of GLUT1, a glucose transporter.
Which is the classic metabolic shift.
It's the classic shift where cancer cells move away from oxygen-dependent energy production in the mitochondria toward glucose fermentation. Seeing that shift, the morphological change in the organelles and the corresponding change in transport expression mapped onto specific hypoxic regions within the native tissue is just revolutionary.
That is incredible context. So HR-IMC is establishing a new standard by capturing protein distribution, organelle-specific signaling events, and these subtle morphological features that directly correlate with function and disease state, all while maintaining that high multiplexity.
And we really have to emphasize this is primarily a technical and computational innovation. It doesn't require new hardware modifications for existing IMC instruments. The knowledge in that custom PSF are the breakthrough.
So while this is clearly foundational, are there still bottlenecks or limitations inherent in this HR-IMC process?
Oh, there are. Because they're working with a destructive process, the limitations right now include imaging speed, laser precision, and, naturally, the signal loss that comes from that destructive oversampling.
But those seem solvable.
They do. The researchers anticipate that signal amplification methods, things like SABR-IMC, could help overcome the signal loss issue. They also suggest future modeling could be refined even further to incorporate subtle physical imperfections, like the Gaussian profile of the laser spot causing crater effects.
Regardless of those limitations, though, HR-IMC fundamentally moves the field. We have moved from mapping large cell populations to mapping the internal landscape of the cell in relation to its hostile environment. This ability to see localized drug effects and metabolic changes, like hypoxia, at the level of the organelle context, could radically reshape how we identify biomarkers and understand the complex mechanisms of drug resistance.
It provides the spatial resolution we need to analyze the intracellular processes that underpin fundamental biology and therapeutic response. It just makes every subsequent experiment higher fidelity.
And that leads us to our final provocative thought for you, the listener. Now that HR-IMC can map the exact location of DNA damage and repair proteins inside a cancer cell and see their colocalization with open transcription sites, what specific, complex drug resistance mechanism, perhaps involving a cell sensing damage and rapidly shuttling an essential repair enzyme out of the nucleus before it can be targeted by a drug, could this level of detail help us uncover next? Think about the movements you can now track that were entirely invisible before.